started here. So my name is Nathan Stevenson, and I'm going to be presenting a little bit of my mineralogy classification of the summit of Mauna Kea as it relates to my overall thesis research goals, mapping optimal vacubug habitat. So there's a couple overall goals for this project. Number one was to find available training data that I can use to create a classification with, and then create this supervised classification and verify the results somehow, and then most importantly, create a useful map in the end. So here's just a satellite image of the summit, and as we can see, there's a lot of variability. There's these different colors and different textures all spaced across the summit, and I just wanted to create a classification that really picked up on these details. So I needed to find available mineral data to start, and there's a whole bunch of it. Back in 1996, Wolf created this mineral map of the entire Hawaii island, but as we can see, the summit isn't very detailed. In order to create a supervised classification, I wanted something just with a little bit more detail. So I found these, which Guinness et al. Created, mapping surface minerals across the summit, but unfortunately the whole summit isn't filled in. There's only a couple small portions where they map these minerals, and I wanted something that really filled in the entire summit. So I kept looking, and I found this, created by Swayze et al. Back in 2002 using AVERIS data, which is something that we talked about a little bit in class, and he mapped the surface minerals present across the entire summit. So great, I wanted to use this AVERIS data, and here's not just the summit, but most of the mountain actually of Mauna Kea here, and this is a 20 meter by 20 meter pixel size. So the question is, does 20 meters by 20 meters accurately pick up the variability and the detail present across the summit? And I would argue that it doesn't do a sufficient job, because as we can see here, there's these different features and these different colors and these different rock sizes, and 20 meter by 20 meter pixel size just doesn't capture this small detailed changes in variability across the summit. So I wanted to produce my own classification using this as training data and apply it to WorldView 2 satellite imagery, which has an increased spatial resolution. Problem number one is they aren't geolocated. Back in 2002, when this AVERIS data was collected, it was before geolocation was implemented, which means that it doesn't have real-world spatial coordinates. It's basically just a picture. So I needed to image-to-image register these, give AVERIS classification real-world coordinates so that I can use its training data. So we do that by selecting tie points. So I zoom way in. This is Keck 1 and Keck 2. I select Keck 2. I go over to my WorldView 2 satellite, Keck 1, Keck 2, and I select Keck 2. And I'm basically saying, hey, this is the same point in these two images. So I do it again. Here's VLBA. I select it. Go over to VLBA, select it. And I do this 50 total times. And then ENVI kind of registers these two images together and gives this AVERIS classification real-world coordinates. So now they are next to each other. They're registered. I got them linked in views. Awesome. I can overlay them together now. As we see, it looks pretty good. Let's zoom in and check it out. You can see that the road lines up with the road. Great. So now AVERIS has real-world spatial coordinates, and I can use it to create a new classification. So in order to do that, we need to take training data, where we'll select our ROIs, choose an appropriate classification method, and then do some post-processing cleaning, and also do some verification. And we can do all this in ENVI. So to select our training data, as we did in class, just like our supervised classifications, we zoom way in, make sure we select the appropriate pixels for the different classes that are present. In my case, there's 6 mineral classes, so I select some red pixels, I select some purple pixels, and I do that all across the summit. And I'm being careful to select a similar amount of pixels for each class, as opposed to a whole bunch in red and not a whole bunch in green. I try to select the same amount. Oh, I'm getting problems again here. So now it's time to pick a classification method, and my overall favorite is a maximum likelihood supervised classification. And this assumes normal distribution of statistics across your pixels, and ENVI then calculates the probability that a pixel is in a certain land cover class, and then it assigns it to that mineral class in my case. So great. This is what ENVI produced as a maximum likelihood supervised classification using AVERIS data. But if we zoom way in, we can see that there's some spurious pixels out here, some just 1 or 2 pixels of yellow and this big expanse of red, and is that real, is it not real? Well, we can apply some majority-minority filtering to kind of post-process clean this image into a little bit better cleaned classification. So that's what I did here. I'll go back and forth here, hang on a second, back and forth quick. So here we go, back and forth. We see these couple spurious pixels, and now those spurious pixels are gone. So we have a cleaned image. Great. So here's my final mineral classification with 6 different surface minerals present across the summit. And so here is, just to recap, a 20-meter Avarice data classification and the increased half meter. I'll go back and forth. So now we can see we're actually picking up some of those small scale changes and that variability present across the summit much better than a 20-meter classification did. So again, that's half meter by half meter. So now we want to justify this since it's new and it doesn't line up exactly, and that's because 1,600 pixels of this new classification fit into a single pixel of the old Avarice data. So it's a little bit different, so we wanted to justify this somehow. So I went up to the summit and I collected cinders at each of these little black X's. I navigated to them using a Trimble, and I come across some cinders like this. So this is hematite, and as we can see, there's a lot of variability. It's not just an expanse of red or an expanse of purple. It's not as simple as my classification on the left. It's actually pretty variable. So instead of just collecting 1 or 2 rocks, I actually collected a whole bunch of surface rocks, and it looked like this, 20, 30 rocks to represent what was actually present on the surface there. And then I wanted to do some ASD spectroradiometer analysis, similar to what we did in class when Ryan showed us the green leaves and the red binders and the yellow leaves. So I wanted to use the similar techniques to analyze this data here. So here I am collecting some spectra data, and what I did was I dumped out most of the bag that we collected onto a black plate that I sprayed so that it wouldn't mess with the spectra of the minerals, and I'd get a clean signal. So I did this for a whole bunch of different samples, 29 in total, and we get some interesting results. So here's just one graph, here's another graph, but if we plot them all together, we can see that they're different. These minerals are different. They have different wiggles and jiggles, they have a different spectral signature, and that's what we use to actually delineate where different rocks are across the summit. And originally what I wanted to do was compare this to USGS standards for hematite and glossy volcanic rock, etc. But my graphs didn't match up exactly with their graphs, and that could be because I did mine in the lab, maybe they did theirs outside using the sun as an illumination source. I collected mine in Hawaii, they could have collected theirs in Colorado or who knows where. So they didn't line up exactly, but I don't think that's because my samples aren't hematite or iron minerals, but I think that's just because of the conditions, and also my samples were created from volcanic action, which could have mixed the signals and stuff like that. So I wanted to also, if I get permission to, I want to do some X-ray fluorescence analysis, which would require me to grind some of the samples, and this would give us an overall more concrete answer. Instead of a graph, it would spit out something like 80% hematite, 20% something else, and then we can use this, and it just gives us a much more concrete answer instead of just a spectral signature that these graphs here. So just to recap, I found available 20-meter Avarice data, and I used this to select my ROI training areas, and I applied them to half-a-meter WorldView 2 satellite imagery to create a supervised classification. I then did some post-process cleaning of that classification, and also I'm beginning to do some lab verification to make sure that my classification is accurate. So if there's any questions, you can contact me at my email, and I'll leave this up for a little bit. But that was my presentation, and thank you.